We continue chapter 14. Now we're talking about Mendel's law of independent assortment. That is, each allele pair independently assorts from all other allele pairs. That's not technically true in every case, but we'll leave that alone for now. Dihybrid cross is a Punnett square that tracks the inheritance pattern of two traits at the same time. In our example, we're going to be looking at P color and P shape. Each one has a dominant and a recessive allele. With P color, yellow is dominant, big Y, green is recessive, little y. With P shape, the dominant is round, big R, recessive is wrinkled, little r. Now, if we crossed yellow round with green wrinkled, and they happen to be homozygous at all of these genes, well, when you crossed homozygous dominant with homozygous recessive, what did we get last time? We got a heterozygote. We crossed homozygous dominant with homozygous recessive, we got a heterozygote. So really none of the rules have changed. So if you did this cross here, you would expect all of the offspring to be heterozygous at both gene loci. That is, they will be yellow round. But what happens when you cross two of these? Well, independent assortment means you get every possible outcome. So here we're crossing two double heterozygotes, and this time we have to use a dihybrid cross Punnett square, a 4x4 with 16 squares in the middle. Now, even though this looks more complicated, none of the rules have changed. What goes on the top are the alleles in the gametes that have been separated. Same thing on the side. What goes in the middle is what recombines at fertilization. Exactly the same thing as the regular Punnett square. So in other words, if you're going to separate the Y's and you're going to separate the R's, then you're going to get one of each in each of these little squares. So the possible outcomes of the gametes would be a big Y and a big R, a big Y and a little Y and a big R, and a little Y and a little R, because they have assorted independently. Same thing over here. And what do? We recombine the letters in the middle. So here we have big Y, big R here, and every other combination that we have. You can look in your book and look at the rest of this. It would be a good idea to look at your notes. It would be a good idea to kind of write this out for yourself so you know how to fill one of these in. Now, I'm not going to make you fill in a dihybrid cross Punnett square for the test, but if we're going to do a phenotypic ratio, there are 4 phenotypes in this offspring. You can have yellow round, you can have yellow wrinkled, you can have green round, and you can have green wrinkled. Okay? Again, look at your book and your notes on this. If these two gene loci independently assort, then what we're going to have is a 9:3 to 3:1 phenotypic ratio. Okay? So looking at the 4 different possible combined phenotypes, you should get a 9:3 to 3:1 ratio in the case of two gene loci that assort independently if you do a cross where they're heterozygous at both. Okay? But again, the rules of filling out a Punnett square are no different with the dihybrid cross than they were before. Now, that takes care of Roman numeral 1 of the notes. Now, Roman numeral 2 is entitled Extending Mendelian Genetics. Now, I need to explain what that means. See, Mendel discovered the basic principles of heredity, but he didn't discover everything. Okay? So what we've described so far are what we refer to as Mendelian traits. Okay? A Mendelian trait is when one trait is controlled by one gene locus. And that one gene locus has 2 possible alleles. One is dominant, one is recessive. In other words, that's what we've been describing so far. Okay? There are plenty of traits that don't operate that way. So we'll go through some of those inheritance patterns. Starting with incomplete dominance. With incomplete dominance, the dominant allele is not completely dominant over the recessive allele, and you do get a blended effect in a heterozygote. The example of choice here, flower color in snapdragons, where the red allele gives you red flowers, the white allele gives you white flowers, but a heterozygote has pink flowers. Incomplete dominance. Another is called codominance. Codominance is when more than one dominant allele exists, and when both are present, both are expressed. Okay? Because that's what a dominant allele is. It's expressed when present. That's how we get type AB blood. Okay? Is that A is from a dominant gene, and so is B. But type O is recessive. That's why we have ABO blood types. Now, because for the human ABO blood group, there are 3 alleles, not just 2. So that's another inheritance pattern called multiple alleles. So really I'm using the same example for codominance and for multiple alleles, our blood types. Now in your notes and on page 273, you have the inheritance pattern of the human ABO blood group. There are 3 alleles. A dominant one for A, another dominant one for B, and a recessive one for O. Okay? So to be type A, you can have the A allele and the O allele, or you can have both A alleles. If you have both dominant alleles, the A and the B, you're type AB, codominant. To be type O is to be homozygous recessive. Study the example. Okay? We also have this in lab. Another inheritance pattern is called pleiotropy. Pleiotropy is when one gene has multiple effects. Most genetic diseases have multiple effects on the body and are caused by a single gene. So that's an example of pleiotropy. Epistasis is another pattern of inheritance where the expression of one gene covers up the expression of another. A good example of an epistasis gene is albinism, being albino. Figure 14.12, page 274, shows you mice. Now here you're looking at another dihybrid cross Punnett square, but this time you're looking at 2 traits, one for whether they're black or brown, and the other whether they're normally pigmented or albino. If a mouse is albino, it doesn't matter what the other genes are. He can't make the skin pigment melanin, he's albino. So the albino gene completely deactivated or covered up the other gene. It is epistatic to the other gene. Okay? Another inheritance pattern is called polygenic inheritance. With polygenic inheritance, one trait is controlled by multiple gene loci. A good example, same page in your book, figure 14.13, skin color. With skin color, it's controlled by 3 sets of genes, not just one set, a total of 6 alleles. The so-called dominant alleles contribute to darker skin pigmentation. The so-called recessive alleles, less pigmentation. So the reason why one of the reasons why there's so many shades is because there's so many arrays of phenotypes and genotypes. See? So with a polygenic trait, you notice that there's a wide variety of phenotypic expression. And even within a family, there can be a pretty good variety. Okay? It is possible for 2 people of intermediate skin color to have children, and the children could be lighter or darker than either one of them, because that's the nature of polygenic traits. In our notes, I also mentioned sex-linked inheritance. These are genes located on the X chromosome. That's a special inheritance pattern of its own. It'll be discussed in the next chapter. A sex-influenced trait is one that is not on a sex chromosome. It's on an autosome, but the gender influences how the trait is expressed. Male pattern baldness is sex-influenced, not sex-linked. It just so happens that the allele is dominant for males, but the same allele is recessive for females. Okay, that's a different inheritance pattern than what we discussed previously. Mendelian inheritance in humans is tracked through pedigree analysis. Page 276 shows you pictures of pedigree analysis. Now the symbols, squares are for males, circles are for females. You shade in the individual that has the trait you are tracking. Okay? Now in general, dominant traits don't skip generations, and if they're autosomal dominant, then you expect to have an equal male-female ratio of those affected by the gene. With autosomal recessive traits, they can skip generations, but they don't have to. And if it's autosomal, you would also expect an equal ratio of male and females having the situation. Examples of recessive disorders include cystic fibrosis, Tay-Sachs disease. Sickle cell disease. Okay? Sickle cell is actually a case of incomplete dominance where a heterozygote has what's called sickle cell trait. They're resistant to malaria, but they don't have the full-blown anemia. Examples of dominant disorders include achondroplasia, Huntington's disease. Now, what makes Huntington's disease kind of unusual is that it's the dominant gene, but it's also lethal. The reason why it can carry on to the next generation is that it has a late age of onset. A person doesn't develop the disorder, and usually until after they've had their children, have had an opportunity to pass on the gene. Study these notes because it's important.